Wow, this is CRISPR. Are we going to succeed?
There's always a chance that you can blow it.
Let's see what happens. So, Roland, everyone says CRISPR is really easy to do, and I was interviewing a very prominent researcher the other day who said to me, any idiot can do CRISPR. Is that true?
I'm not sure about any idiot, but it is easy, and we're gonna find out. So what we want to do is we want to knock out a gene in a human cell line.
Yeah, I chose a gene that putatively is involved with the immune response to dengue virus. And the reason I chose it is because there's a theory that if you've had dengue virus and you have an antibody to it and then get infected with Zika, it helps the Zika virus copy itself, which might be responsible for the damage that we're seeing in babies. So you and I are going to solve that problem, right?
Hopefully. I mean,
what are the basic steps that we need to do to get started?
first we'll have to define the genomic region that we want to cut. Then we take that information and put it into a design software, in a CRISPR design software. And the third step is ordering the oligonucleotides for the cloning of the guide RNAs. Then we have to order a plasmid.
What's the name of our plasmid?
It's lenti-CRISPR V2.
And we're going to put in here our 20 nucleotide sequence that's going to guide us to the portion of the gene that we're going to knock out.
Exactly.
So we've got the oligos and we've got the plasmids ready to go. Now we're going to pipette
Now we're going to pipette everything.
We're going to pipette it all together.
Right.
Okay, let's do it.
So first of all, we take a tube. This tube is sterile. Now, see, like we already have the problem, we don't know what a tube is, right? So this is like actually a microcentrifuge tube, right?
Okay.
It's a 1.5 milliliter microcentrifuge tube.
All right.
This is where we'll set up our reaction. So the components we will need is the plasmid,
Right.
the enzyme.
Got it.
The enzyme only works in a specific environment, so we'll also need the buffer, the correct buffer. And we also make it like a feasible volume, so we'll add water to it. Go down, push the bottom down, and up.
Just one time?
So, so here's the deal. Hold on, John. Like, can I take the pipette? You were way too fast, so you had bubbles in there. So let's try it again.
So I already ruined the experiment, basically.
Basically, by not knowing how to pipette, you're already like on a bad path, right?
I'm on a
It don't
bad
not
path.
fail necessarily.
Right.
It's very
Okay.
robust,
Okay.
but it's suboptimal.
All right.
All right, here we go again. That looks good. Okay,
All right.
good. Very nice.
Easy peasy. I didn't know it was so much fun to do.
It
CRISPR.
is. And now let go of it. Yes, that was good. That was proper pipetting. Oops, yeah. I think now you kind of like you failed a little bit, probably because you put it like, see like how much liquid there's still left. Yeah, it should be fine. You know, let's keep going. And now you vortex again.
It's a little nerve-wracking. Yeah, I don't want to make mistakes.
Woo!
I'm vortexing.
You're totally vortexing
Totally
right
vortex.
now. And then you spin it real quickly. That's it.
That's it.
Goes on 37. Let's go
Where's
over
the
there.
water bath?
I don't think we need to videotape that one, but.
In the beginning, I said, "Is it really easy? You know, can any idiot do this?" Yes, but then, like, I got a sense of like there's some pride that no, you've got to really be kind of skilled to do it.
I think there is a certain skill level. It's very necessary, right? Like if you take a random person from the street and say knock out CD32 in THP cells, in like this million cells, there is no way this person would do it, right? Not in 100 years.
It's really easy for people who already know how to do what?
To do molecular biology. We just discussed, pretty much everyone knows about DNA. Go to RNA and most people are like, not so sure, right? Mention oligos, plasmids, cloning and so on, you will lose everybody.
Yeah.